Hello everyone, my name is Jinhao Wang. I'm a PhD candidate of chemical engineering in Texas A&M University College Station. Today, I would like to give a presentation titled with Application of Isothermal Calorimetry for Inherently Safer Reactor Design. Before I start, I would like to briefly introduce myself. I got my bachelor's degree in chemical engineering in 2015 at Rutgers University. In the same year, I joined Texas A&M University to pursue a PhD degree in chemical engineering, and I will graduate in 2020. While doing my PhD, I was able to do two internships. The first one is with the Dow Chemical Company as an EMPS intern in their process development group of quality department. Then I was on a 6-month internship as an academic contractor at Eli and Company in their small molecule design and development group. Here's the outline of today's presentation. I will start by introducing the background and objective of this project. Then I will discuss the experimental and theoretical method I use for this project. Then I will share some of the results on experimental data analysis and single factor analysis, as well as more advanced response surface model analysis to explain some of the findings from this experimental work. Then I will conclude the presentations and share some insights of the future work. Now, let's start with the introduction part. If we look at the past incidents involving batch or semi-batch thermal runaway incidents, like T2 incident for example, a normal thermal runaway scenarios can be shown on your right-hand side, the thermal runaway scenarios chart, where the reaction was desired to be performed in a normal processes in green curves. However, when there's insufficient cooling design, significant reaction heat will accumulate inside reactors and cause reactor temperature rise through the blue curve, all the way reaching maximum temperature of synthesis reactions. A lot of time, there could be a secondary thermal runaway reactions, for example, decomposition of ammonium nitrate or decomposition of the methylpurine N-oxide. And those reactions will generate significant heat as well as non-condensable gases. And those heat and gases will cause reactor overpressure, particularly if the reactor pressure relief system was not in place, like what happened in T2 incidents. Complete rupture of reactor may happen and will cause very severe consequences. For T2 case, 4 people were killed and the local community was forced to evacuate. T2 incidents is not a single case. There are a couple more incidents on your left-hand side. CSB has been identified some of the common root causes for those incidents, and those root causes involves insufficient cooling design as well as the pressure relief capability design. And people are not aware of some of the significant reactive-chemical hazards involved in the secondary reactions. And those lessons has been learned. However, incidents like that still happening nowadays. To prevent those incidents from happening, it is really necessary to identify the inherently safer operating conditions in lab scale reactor before the reactor go to full scale or industrial scale. For example, for lab scale reactor like RC1, which reactor size is around 1 liters. However, for industrial scale reactors, the reactor size is at least 1,000 gallons. And the consequence of thermal runaway of larger scale reactors with more hazardous chemicals stored could be more catastrophic. For lab scale reactor like calorimetry, one of the important cell parameters are very important to be identified before we go to full scale. And those parameters include heat-up reactions, and maximum reaction pressure can be reached in the synthesis reactions as well as the thermal runaway secondary reactions. And what's the maximum temperature can be reached during your synthesis reactions. And how much catalyst you're supposed to load to reach safer conditions as well as the higher efficiency. And what's your raw material feed rate, for example, if you have a semi-batch reactor? And what's the optimal condition to reach maximum product yield, which is very important for efficiency and economic perspective. And there are many more others can be done using calorimetry and in lab scale reactor. Today's presentation will mainly looking at a case study of thymethylpyridine N-oxidation reaction via hydrogen peroxide. We are trying to study this reaction and trying to see how we can use calorimetry and lab scale reactor RC1 to identify the important critical operating parameters like reactor pressure and enthalpy, and so as to help safer and more efficient operation of full-scale reactors. This chemistry, N-oxidation reactions involving two reactions. The first reaction is a main reaction, which hydrogen peroxide will oxidize the methylpyridine to generate methylpyridine N-oxide and water, catalyzed by the catalyst called phosphotungstic acid. However, the catalyst phosphotungstic acid will also trigger the decomposition of hydrogen peroxide, which will generate water and oxygen. There are several hazards involving these typical reactions. The first hazard involving this reaction is that both reactions are highly exothermic. For main reaction and oxidation reactions, the reaction enthalpy is around 200 kJ per mole, and the decomposing of 1 mole of hydrogen peroxide will generate 100 kJ or more of enthalpy. The second hazard involving those reaction systems is the final product, thymethylpyridine N-oxide, will decompose at temperature about 200 degrees Celsius, which will generate significant amount of heat as well as the non-condensable gases. Meanwhile, as the oxygen was generated by the decomposition of hydrogen peroxide, which will increase the reactor pressure significantly. The third hazard involved is generation of oxygen, which not only will trigger reactor overpressure because it is non-condensable, and also in the presence of flammable chemicals like pyridine, it will cause flammability issues and fires and/or explosions. To summarize, if you look at the thermal runway scenario chart, where in the normal processes for an oxidation reaction, you have a highly exothermic N-oxidation reaction as well as in parallel decomposition of hydrogen peroxide, which will generate lots of heat and oxygen. When temperature is about 200 degrees Celsius, which will cause the decomposition of the final product and oxides, as well as more catastrophic decomposition of hydrogen peroxide. So this reaction is very hazardous as well as has important application in pharmaceuticals as well as fine chemical. Typically, this reaction is performed in a semi-batch mode in industrial practices, where hydrogen peroxide is gradually dosed into the reactor, which contain catalyst as well as the thymethylpyridine, under a temperature range of 95 degrees Celsius to 100 degrees Celsius. The objective of this work is trying to look at the critical design parameters of these reactions, which is the catalyst amount you load, and the reactor temperature, as well as the hydrogen peroxide dosing rate, such as to minimize the reactor pressure due to decomposition of hydrogen peroxide and maximize the N-oxide product yield, and how we can play with these three parameters in RC1 calorimetry and able to achieve both efficiency as well as the safety objective. Now let's talk about the methodology we employ to achieve our goals. First, let me introduce the experimental method I'll be using, which I'm using RC1 reactor. As you can see on your left is a schematic representation of the RC1. So this RC1 is isothermal semi-batch reactor, which allows synthesis of N-oxide at different temperature and hydrogen peroxide dosing rates, as well as different catalyst amount. And hydrogen peroxide in the can be gradually dosed through a dosing pump inside the reactor, which contains the catalyst and the methylpyridine. And during reactions, there's a FTIR spectrometer was inserted, which monitors the reaction species concentration changes. And meanwhile, the reactor pressure was kept monitored, and the reaction enthalpy and heat flux was able to capture by the heat flux sensor band. And the reactor was able to control the stirring rate as another variables, and the maximum temperature, maximum pressure allowed inside the reactor is 10 bars. After the reaction finished, we were able to take a sample products and perform HPLC, GCMS, as well as the RSM test to identify the major compounds and the side reactions. Now let me introduce the theoretical method I'm using, which is response surface methodology. Response surface methodology is a statistical method which was able to quickly identify the relationship between input variables and output variables without detailed understanding of the system capacity. It consists of 4 steps. The first step is the variable screening. For this case, as I mentioned in the objective part, we are looking at how catalyst amount and temperature and hydrogen peroxide dosing rates will affect the reactor pressure as well as the product yield. And based on those 3 parameters, we perform design experiments and trying to identify the minimum number of experiments required to understand the system complexity and the relationship between input variables. We were using box beacon design experiment. And then we perform mathematical and statistical treatment to get a regression model which correlate the input variables, catalyst, temperature, hydrogen peroxide dosing rates with the output response, such as the reactor pressure as well as the product yield. And based on the regression model we obtained, we do system response surface analysis, which allow to optimize the reaction conditions and identify the inherently safer and more efficient operating conditions. Here's the design experiment chart we employed. As I mentioned, we were using box beacon design experiments on your right hand side, which allow you to perform 3 variables at 3 levels, and within 13 experiments you're able to map the all design space. More specifically, we are running reaction under low temperature of 85 degrees Celsius, medium temperature 107.5 degrees Celsius, and high temperature 130, and same for catalyst, low for 4 grams, medium for 8 grams, and high for 12 grams. And dosing rates, low for 2 grams per minute, and medium for 4 grams per minute, and all the way to the high dosing rates, 6 grams per minute. For each experiment, we perform twice, and by the end of the reaction, we measure the product yield, as well as require the final reactor pressure as shown right here. As you can see, under different combination of a temperature, catalyst, dosing rates, we have significant variations of different yields obtained, and as well as different reactor pressure we obtained, which means the 3 variables we chose, the temperature, catalyst, dosing rates, has very significant impact on our reaction system. Now let's look at the data analysis part first about the calorimetry data. So here's the typical data for 170 degrees Celsius with the 12 grams catalyst and 2 grams per minute hydrogen peroxide dosing rate. Let's look at the right curve, which the reaction enthalpy change rate first. We start with the calibration step, which identifies the heat transfer coefficients. And then reaction, the whole system stabilized, which is able to obtain the baseline value for reaction enthalpy generation rate. And when reaction starts by dosing of hydrogen peroxide in the black curve, as you can see in the black curve, the reaction volume is increasing due to additional hydrogen peroxide solutions. The reaction enthalpy quickly goes up and still stabilizes at a constant value, which indicates the reaction was controlled by the dosing rate of the hydrogen peroxide. After dosing stops, the reaction enthalpy quickly goes back to the baseline. After stabilized, we did another heat transfer coefficient calculations through calibration. If you look at the green curve, which is the reactor pressure, similarly, before reaction starts, we have a very constant flat reactor pressure profile. After the reaction was initiated, we were able to see the reactor pressure gradually increase due to decomposition of hydrogen peroxide. And the reactor pressure finally reached a level around the 3 to 3.5 bars after reactions. And if you look at the reactor temperature profile, and by changing the jacket temperature in the blue curve, we were able to perform reaction under isothermal conditions throughout the reactions. Now let's look at the FTIR data. As I mentioned before, we have an infrared spectrum spectrometer, which allows us to measure the infrared spectrum of the reaction species every 48 seconds, which gives us a 3D surface of FTIR data, where the x-axis is the wave number, and the y-axis is reaction time, and the z-axis is the peak height. As we can see, some of the peaks are increasing over reaction time, indicates they might be reactants, and some of the peaks are reducing over time, indicates they are the products. And this 3D surface actually it allows us to visualize how the functional group peaks are changing over reactions. Now let's compare the infrared spectrum before and after reactions, where the before reaction is the blue curve and after reaction is the pink curve. And we were able to identify some of the distinct peaks that belong to product, and some of the peaks belongs to the reactant. If we plot the peak height change with time of those distinct product and reactant peak heights, and we were able to see the peak height of the reactants are reducing over reaction time. However, the peak heights of the products are increasing over reaction time, which makes sense. After we do the normalization of those peak heights, what we are able to see that those distinct peak heights of the products and the reactants are actually overlapping together, which indicates they belong to the one compounds. And those information allows us to do more quantification analysis and even obtain the concentration change of the products and the reactants. Apart from using infrared spectrum analysis of the transient concentration change over time, we also analyze our final product using GC. As you can see, there are only two distinct final products shown here. One comes up at 5 minutes, the other one comes up at around 13 minutes. After going through the max spectropy as shown here, we were able to identify a good match of the reactants and the products in our MS results. Indicate the reaction selectivity is actually pretty good. All the final product, the methylpropylene oxide, were generated. Similarly, HPLC results are shown here. We were analyzing our final products and give us a product peak and reactant peak, which is not surprising and meets with the results from both FTIR as well as the GCMS. As you can see, at different reaction conditions, the peak height of the products and the reactants change significantly due to the different product yield. Now let's go to this single factor analysis. The idea is trying to do control experiments, trying to see the impact of each factor, temperature, catalyst, and dosing rate, and how their how they will change the reactor pressure as well as the product yield. So here's 4 groups of control experiments which we are running at same temperature and same dosing rate while different amount of catalyst, which is 4 grams and 12 grams. As you can see, at whatever temperature dosing rate condition apply, higher catalyst amounts will always allow you have a lower reactor pressure, which means high catalyst conditions actually favor the main oxidation reactions. Similarly, and not surprising, we are able to see high catalyst amounts will give you a higher product yield compared with lower yields obtained at the low catalyst amount. Now let's look at the impact of temperature on reactor pressure and product yield. So here's 2 control experiments which we are fixing the reactor hydrogen peroxide dosing rate and the catalyst amount loaded. However, we are changing the temperature from low degrees Celsius of 85 all the way to the high temperature 130 degrees Celsius. As you can see, at higher temperature, we are able to actually obtain lower pressure, which seems to violate the physical law. However, since the high temperature condition favor the main oxidation reaction and inhibit the decomposition of hydrogen peroxide, this is not surprising at all, because we are able to have a higher selectivity and reduce the decomposition of hydrogen peroxide and oxygen amount, which lower the reactor pressure. Similarly, at the N-oxide yield, we are able to get higher yield at higher temperature, and which also fit the results with the reactor pressure profile. Now let's look at the impact of the hydrogen peroxide dosing rates. Essentially, higher hydrogen peroxide dosing rates will give you a higher concentration of hydrogen peroxide inside the reactor. So here are 4 group of control experiments which compares the impact of low dosing rates 2 grams per minute and high dosing rate 6 grams per minute under same temperature and catalyst amount. As you can see, employing higher dosing rates will give you a higher reactor pressure due to more hydrogen peroxide decompositions and more oxygen generated. However, under condition where you have a high temperature or high catalyst amount in the figure C and D, changing the concentration of hydrogen peroxide or dosing rates will not impact the reactor pressure that much. If you compare the product yield under low peroxide dosing rate and the high peroxide dosing rates, similar to pressure profile, we are able to obtain lower yield at higher dosing rates. Based on the input temperature, catalyst amount, and dosing rate, we were able to build regression model correlating the 3 variables with the reactor pressure and the product yield respectively. And here's the equations, our quadratic equations for correlating the input temperature, catalyst, and dosing rate with the output pressure and yield. We were able to get very good statistical test results and passed the significance of fitness as well as the lack of fit. Now we perform response surface analysis by visualizing the regression model we developed. First, let's look at the impact of the catalyst and temperature on the reactor pressure and the final product yield. So here's the pressure response contour in terms of different catalyst and different temperature. As you can see, under conditions of high temperature and high catalyst, you were able to have a lower reactor pressure, which is very corresponding to the results obtained from single factor analysis. Similarly, when we perform when we vary the catalyst amount and the temperature, as we can see, in the high temperature and high catalyst amounts, you are able to get more efficient zoom. And in this case, by comparing the pressure response contour and the N-oxide response contour, we were able to see that actually safety and efficiency can be achieved simultaneously. We are able to have a low pressure as well as the high yield under high catalyst amount and high temperature conditions. Now let's look at the impact of the catalyst and dosing rate together. We were able to obtain a response contour for pressure, where under high catalyst amount and low dosing rate, we are able to have a region of low pressure of 4 bars. However, when we have a high dosing rate and low catalyst, the reactor pressure can reach about 12 bars. As you can see, comparing the response contour for pressure and N-oxide yield, a similar shape was obtained as well. High efficiency region and lower pressure region were obtained under high catalyst amount and low dosing rates. Similarly, and not surprising, we look at the impact of the temperature and dosing rate together. Under low temperature and high dosing rate condition, you have a pressure of 22 bars. However, in the opposite, when your temperature is high and your dosing rate is low, you will achieve high efficiency of the N-oxide yield as well as the safety performance of the low reactor pressure simultaneously. We also validate our response surface model as shown here, where by validating our model using new experiments performed in this study, as well as experimental study in literature as shown in this table. As you can see, the predicted reactor pressure and yield match with experimental results pretty well, indicating that our model can represent the system in a good way. Now let's concluding our presentation. We were able to see that the main oxidation reaction favor higher catalyst amount, temperature, and lower dosing rate conditions. Meanwhile, by using the response surface methodology, we were able to find that safety, which means reactor pressure is low, and the efficiency, which means we achieve high final product yield. And these two objectives can be achieved simultaneously under intensified reaction conditions, essentially under condition where you have a high temperature and high reactant concentrations. For the future work, we will be doing first principle kinetics and thermodynamics model, which is actually under review by ASHE journal. We were able to build a model to predict the concentration change as well as the reactor pressure change over time. And we were also planning to use this model to do infinitely safer reactor design through parametric sensitivity analysis. And by the end of the day, we are trying to design a continuous reactor to perform these reactions and perform the safety and economic analysis of these two types of system. This work towards identification of intensified reaction condition using response surface methodology, a case study on the methylpurine N-oxide synthesis, has been featured on the cover of special issue for framework for process intensification and modularizations on journal Industrial Engineering Chemistry Research. For more information, please refer to our publication. This concludes my presentations. Questions and comments are welcome, and feel free to send me email through this address. Thank you.